Okay, I think we're right at the top of the hour, so welcome to another CBR seminar. I'm going to ask Ed Conway to please introduce today's speaker, but welcome, Dr. Licht.
Thank you.
So it is my great pleasure to, and you'll excuse my raspy, Dana calls it sexy, I just call it a cold voice. But it's really a great pleasure to introduce Christoph. Christoph completed his medical training in Freiburg in Germany and did a variety of stints in his training in nephrology and pediatrics mostly in Cologne, and then at the University of Texas Southwestern in Dallas, and landed, fortunately for the Canadian community, scientific and medical community in Toronto at the Hospital for Sick Children and the Sick Kids Research Institute in 2006, where he has risen through the ranks appropriately for all his contributions. And he is now a pediatrician, professor of pediatrics at U of T, and is a senior scientist at the Sick Kids Research Institute and in the division of nephrology, or pediatric nephrology. Christoph has a story, a really famed history in the field of complement, particularly as it applies to pediatric diseases, and that in particularly relates to atypical aHUS, but other complement-mediated disorders. He really was involved in groundbreaking work in the introduction of at the time, the only drug that was useful for treating atypical aHUS. And that was highlighted in many publications that Christoph has been involved in. But he's not only is he a fantastic clinician, but he's really a clinician scientist with a very fruitful and fascinating and really creative interests in the role of complement, particularly as it pertains to the endothelium. And I've had the pleasure and honor of collaborating with him most recently in the last year or so. Christoph's obviously been recognized with many awards, lots of great grants and numerous publications, and we're really pleased to have him here. So Christoph, it's all yours.
Thank you so much, Ed, for this extremely kind introduction, and also for the invitation. It's a pleasure to speaking to this crowd. Unfortunately, I can't be there in person, and I hope for kind of another opportunity in the near future to see all of you in person. Since I was really given not much guidance, which I totally appreciated, in terms of the title, I thought the best I can do is give you a little bit of an overview talk of what we are doing, in the lab, and called the talk "Translational Research in Complement-mediated Diseases." I will focus, as you can see very soon, on the TMA aspect of this spectrum of diseases. So I'll start with an overview on complement and disease in general, and we'll then go into an interesting aspect that we were working or continue to work on, which is the contribution of neutrophils in the pathogenesis of TMA. And then want to share with you a couple of fascinating findings in terms of how endothelial cells respond to complement attack, both short and long term, and then try to wrap this up. I have a couple of pictures throughout the talk for you guys in BC. And actually for all Canadians, it's probably not really a big thing, but for me, with my German background, it's still just overwhelming to see how beautiful your country is, this country is. This is from Lake Manitou on Manitoulin Island that we had the pleasure to visit in early summer, actually this year, for a long weekend. Okay. Complement and disease. So this talk doesn't work without at least a high-level introduction into the complement system. As you many of you might know, it's part of our immune system. It belongs mainly to innate immunity, and its major job is to distinguish between self and non-self. It's an interesting and I find fascinatingly organized biological system that depends on its balance between activation and inactivation, very much like the coagulation system. And activation is actually not a bad thing. It's actually needed at the right time and at the right site to defend the host against invading microbes. But when the activation is not regulated towards the right side and time, but is uncontrolled, then damage occurs and diseases arise. If you go into a more detailed level and look at the complement system in terms of its components, protein components, and their functional connections, then you have to work through something that looks like this, which is basically an overview of the major components of the complement cascade. This cartoon is helpful since it shows you in one shot that there is proteins that in a domino fashion work in sequence to activate the next step, to end up starting from a starting point, end up at a terminal effector. The starting points are called the classical, the lectin, or the alternative pathway starting point. Classical pathway initiated by the presence of immune complexes, lectin pathway by certain repetitive carbohydrate sequences on the surface of bacterial envelopes, and the alternative pathway differently from that, like endogenously or constitutively active. Those three pathways merging in the activation of the key complement component C3 to kick off the steps of the terminal pathway all the way to the formation of this MAC, membrane attack complex, or C5b9. In those lighter kind of brown spheres or circles, you see blockers. Unfortunately, my pointer does not show, so I have to really do it with my words. You see that those steps, and you see those red kind of T's indicating blockade. So those are the regulators that are built into this system from the top to the bottom to keep it tightly controlled and really allow activation only where and when needed. As I said already, when this regulation is out of control, disease arises. And the current concept is we started from a very limited number of conditions and situations of dysregulation to now a concept that includes local and systemic dysregulation of complement, acute and all the way to chronic, and then various forms of dysregulation of the complement you see in the middle in those boxes. And all I want to do here is to show you this spectrum of conditions and organ systems that can be involved by these various forms of complement dysregulation. The two that are really relevant to a nephrologist, there's more, but the two that are really at the center of nephrologists being interested in complement-mediated diseases are one type that affects the homeostasis of the endothelial cells, which is what we call thrombotic microangiopathy. And the other group is what we call MPGN or C3 glomerulopathy, where deposits are placed into certain spaces in the glomerular filter, which the left side is a cartoon of. of the kidney. The diagnosis, the specific diagnosis, are called IC-MPGN, C3 glomerulopathy, or dense deposit disease, which we're not talking about today. But we are talking about this phenomenon of the, or the diagnosis of the complement-mediated thrombotic microangiopathy, or atypical aHUS, which is the condition, as I said, where complement dysregulation, again, in this blood vessel here, another rendition of the complement cascade, leading to the formation of those C5b9 complexes in blue, a little to the right at the bottom, resulting in injury of the endothelial cells, resulting in the formation of clots, the consumption of platelets and red cells, and with that, dysfunction of the dependent organ system, downstream of the clots that prevents proper blood supply and oxygen and nutrient supply. The learning over the past few years has been that, again, from very, very narrowly defined conditions, we are now understanding that there is a spectrum of TMA diagnoses that all have their own core pathology or disease driver, if you want. Besides what we call the primary atypical aHUS, that is the ones where we have complement-related genetic or autoimmune dysregulation. That's the clean forms of complement-mediated TMA or aHUS. But then you have this purple box on the left side, where you have all sorts of diagnoses or conditions or drivers, including drugs, infections, autoimmune conditions, malignancies, hypertension, various conditions that in the end of the day can present or result in a clinical picture of a TMA that is indistinguishable from the primary aHUS. And really the key is that for the time of the active disease, the complement system is temporarily recruited to enhance the damage, as you can see in this cartoon. So it's a non-complement induced, but then complement system enhanced situation of injury to the endothelial cells. And with that, and that is a talk in its own right, complement blockade for those secondary forms of TMA can at least be justified or considered for a limited period of time. Let's slowly move to what I promised, which is kind of the work in the lab. At some point, we just wanted to visualize what happens to an endothelium that is under complement attack. And this is now endothelial cells, a monolayer. On the left side, it's an intact monolayer. In blue, the nuclei. In green, some sort of fibers in the cells that identify kind of where the cell body is, and then in red, the weak adherin to outline the border of the cell. And as you can see on the right side, under complement stress, you see at least two major differences. One is that those cells look upset, disrupted. The actin cytoskeleton the fibers are less nicely organized compared to the control, and the cells lose contact. You can even measure that if you measure the transendothelial current or kind of resistance. You can measure the difference with and without complement damage. We know obviously clinically quite a bit how to handle these situations. We have a textbook understanding of complement attack injures and eventually destroys endothelial cells, but there is a number of unanswered questions One way to kind of rationalize that you continue to work on this phenomenon of complement attack to endothelial cells is to make yourself aware of the fact that not all of those situations are explained by mutations in antibodies. So there must be other causes. The penetrance of these mutations in antibodies is incomplete, 50% only percent, and that's probably a very kind of poorly informed number or percentage. As is the time and age of disease onset, which is quite unpredictable. And then very obviously our response, the endothelial response, our understanding of the endothelial response is quite incomplete. And the concept of cells, immediately and dying from complement attack is misleadingly simplifying. So you can put into a hypothesis. We were interested in what else is there to understand and to learn. And to study that, now I will show you a few, I think it's three stories from the lab, how we have tackled that. And I started by recognizing the student in the lab who has led the work. In this case, it was a PhD student, Magdalena, who is at the moment a clinical fellow with us. So for the work that we're doing for this particular project, but also in general in the lab, we had to generate an in vitro / ex vivo system that allowed us to study our questions. And we landed on this, on a bioflux microfluidic system where in those microchannels, endothelial cells can be seeded and can be perfused, mimicking what is happening in the blood vessel. And not only the composition and the resulting composition of the perfusate, but also by a microscope, what's happening to those cells can be monitored. The second challenge that we had to address sounds simple, but took us the better part of 6 months. Or took us 6 months really to resolve, was we needed a system that allowed us to basically stress these cells with complement. You find in the literature various protocols for that, and in our hands, neither a protocol like Cymosan or copper factor, nor just complement serum incubation worked to in a controlled fashion activate complement on endothelial cells. What we had to do was we had to sensitize the cells with an antibody against an antigen found expressed on the endothelial cells. We chose CD59, and then provide serum as the resource for complement proteins. So that's the first line of the table at the bottom. That is our basically main protocol for complement activation. And as negative controls, we used heat inactivated serum or various depleted sera, including C5 depleted serum. We documented that this really resulted in complement deposition on the cells, as shown here by flow and C3 deposition with the red peak. And here in an IF study documented that not only C3 was deposited, but also C5b9. So the cascade was really completing to its final step. As you see, the red staining on the right side. And then our question was really, is there are there other cells besides the endothelium and its responses that might be involved in what's happening in a TMA situation. So what we did was we looked at the endothelial cells in this microfluidic channel system. Here's two channels, one at the top, one at the bottom, and perfused them with serum. At the bottom, normal serum, so that is our complement active situation, and at the top with depleted C5 depleted serum, which is our negative control, and non-activated neutrophils. And when we perfused those neutrophils, the white spots, you see them adhering to the complement activated endothelial cells when complement is active, but not when complement is not active at the top. We quantified this. This is basically the bar graphs representing what you have just seen. We have not only used C5 depleted serum as a negative control, but also C6 depleted serum. And then we co-perfused endothelial, the same system with the complement activated endothelial cells in the microfluidic system with neutrophils and platelets. And again, we made sure that these platelets are resting and not activated. And we found, not really to our surprise, but interestingly, we found that the platelets adhered to the endothelial cells at the sites where the neutrophils were adherent. So what we felt we were looking at here really was the formation of a clot. Since we're doing this in the absence of serum, of plasma, I'm sorry, I can't really call it a clot. It's an aggregate of platelets and neutrophils, but it was a phenomenon that really was mainly or almost exclusively occurring at the sites where neutrophils were adherent to the endothelial cells, which is published, and we claimed that basically we with this experiment identified another player in the formation of complement-driven clot formation in TMA, which is the neutrophils. We tried to test this now the other way around. It's in the same paper, where we said if that is true, then in patients in whom who are treated to basically block their complement system, we should see less neutrophil adherence to endothelial cells, and that is shown here on this cartoon, where we have aHUS patients. This is a sample, a small sample size that we had at hand. So this is not kind of a major randomized study, but still 5 patients with different conditions leading to an aHUS or complement-driven TMA phenotype and various types of treatments. We have no treatment, patient 1, we have plasma infusion for patients 2 and 3, and we have eculizumab treatment for patients 4 and 5. And the bar graphs represent the neutrophil adherence to the endothelial cells. And you can see that in the patients with eculizumab treatment, where you efficiently, different from the plasma infusion, efficiently block the complement system, you prevent neutrophil adherence to the endothelial cells, which is in keeping with the treatment success that you achieve with eculizumab in these patients. So, summary 1, we found that neutrophils adhere to complement-activated endothelial cells, and we identified that these adherent neutrophils provide a nidus for clot formation in the context of a complement-driven TMA scenario. We feel that this is very much in keeping with what is very actively discussed in the field, and we published in this cartoon 2014 already as well, which is a double or multiple hit basically manifestation event or process in TMA that requires or combines prevalent risk factors and additional trigger factors. So patients may have no or mild or severe underlying susceptibility factors, and if they had any, it could be genetic or autoimmune, on top of which they will accumulate triggers, be it infections, be it other stressors. And at some point, the combination of these phenomena will result in the crossing of the manifestation threshold, below which you will not see symptoms of the disease. And obviously, treatment is not undoing either the underlying condition or the trigger, but it is just raising the bar to preventing manifestation that way. A little break. Same place, Manitoulin Island, Lake Manitou. And off to the next story. So, having contributed this, extension of the manifestation of TMA as a disease, we then really turned to the response of the endothelial cells, and this was driven by basically the observation, and I mean also some literature basically pointing out that this is not a pathway of no return, as suggested by some textbooks. The endothelial cells are obviously in a position where they can recover and not only surrender to any complement attack, but basically what and how is that happening was unknown. So as I showed you in the very beginning with introducing the complement cascades, the end product of the activation sequence is the formation of this membrane attack complex, the pore that basically perforates the phospholipid bilayer and allows for multiple things to happen, but one major consequence is following the concentration gradient, the calcium influx into the cells. And we documented that with this pseudo-labeled calcium stain of endothelial cells, that basically the moment you activate complement on the endothelial cells are flushed with calcium, which then results in this bright light signal that you can see here on this video in loop. We quantified this result, and as you can see here, achieved this immediate upstroke in terms of calcium influx into the cells upon complement activation, which is our sensitization protocol in red, comparable to what you see in gray, our positive control calcium ionophore, and as opposed to the negative controls heat inactivated and C6 depleted serum. We repeated, or as I said, one phenomenon that occurs is the calcium influx, But the other phenomenon is that those cells obviously leak, content of the cells. And what we looked at here is we preloaded the cells with calcein, and then over time, and this is a time course of 60 minutes, basically followed in the supernatant what leaked from the cells. And interestingly, we saw an expected increasing amount of calcein release over the first 20 minutes, but found a plateau afterwards. So after 20 minutes moving forward, there was no leakage anymore. And as you can see with our negative control heat inactivated serum, there was no leakage at all. And then we asked the question the other way around. What happens now if we challenge the cells with something that would come from the outside and stain the inside of the cells? And we use this dye that stains intracellular membranes in general, FM1. As you can see here, a negative control with an intact cell membrane, you wouldn't see any staining inside the cell. So that's the left picture, and the right picture would be a positive control, the saponin perforated cell membrane that allows for the influx of the stain and the highlighting of those intracellular membranes with the green, strong stain. And then we applied our complement stress protocol to the endothelial cells and did a time course over, again, an hour, and found, in keeping with the calcium results that I've just shown you, similarly that the influx of, in this case, the dye into the cells stopped after about half an hour. So after 60 minutes, this is obviously not the same cells. This is a different independent experiments, stopped at 5, 20, and 60 minutes. The influx of the dye stops at some point. So, and this is just quantification. So what we hypothesized with that finding was these cells are injured, there is perforation, there's in and out, calcium in, and we showed the calcium out. But this seems to be repaired over time. And the hypothesis then was that this would allow the cells to survive. So the question was, what could be a mechanism of repair? And the hypothesis we came up with was that this could have to do with something that is very actively happening in endothelial cells under stress, which is the release of von Willebrand factor, which is obviously contained in the endothelial cells in viable Palladi bodies, and upon activation, one of which is complement, endothelial cells release their content of von Willebrand factor by shuttling the viable Palladi bodies to the cell membrane and releasing that way merging the viable Palladi bodies with the cell membrane, and that way releasing the von Willebrand factor. So we have published this already in the past, but we repeated it here to show in a time course experiment over 30 minutes that von Willebrand factor in red, stained at the outside, is released from the inside to the outside of a cell, and at the same time, the intracellular pool in green in the middle column is depleted. Not completely, but to a major degree if you compare the 10-minute and the 30-minute middle column result in green. And then, following our the knowledge in the literature, but also our hypothesis, at the same time, you find the appearance of CD62, P-selectin, which identifies the viable Palladi body membrane appearing at the surface of the endothelial cells, which is the red dots in the right column that are not present at the early time point, but in high amount present at the 30-minute time point. So the hypothesis was it is the release of von Willebrand factor via fusion of Palladi bodies with the plasma membrane to facilitate endothelial cell resealing. So we wanted to test this. We used BOEC cells. I'll skip through to save a little time. We can talk about that as a tool a bit more if you want. The reason that I need to introduce it here is to share with you the fact that we were able to use endothelial cells for the next experiments derived from patients with certain genetic conditions. So this is not in the first place manipulated endothelial cells, but it is cells generated from patient donors. And the patient donors that we chose were type 1 and type 3 von Willebrand disease patients. So you are more hematologist than I am, of course. So my understanding, our understanding was in type 1, you have reduced amounts of von Willebrand factor, but key is you have normal Palladi bodies, and in type 3s, you lack the von Willebrand factor and the Palladi bodies, as seen here on Western blot and mRNA level on the right side. And then we repeated using now the type 3 von Willebrand disease patient endothelial cells. We repeated this calcium release experiment. So endothelial cells stressed with our complement activation protocol, and we measure basically what is leaking out of the cells. In the white open bars, you see basically what you've seen already, that the release is basically plateauing after 20 to 30 minutes. If you do this experiment, if you repeat this experiment now with the type 3 von Willebrand disease BOECs, you see the release continuously increasing over time, being in keeping with our hypothesis that these cells, as we would be expecting now, were not able to reseal with that repair their membrane. And then we basically tried to really nail it by retransfecting, or not but by transfecting the type 3 von Willebrand disease BOECs to express GFP-labeled von Willebrand factor. And you see now here the repeat experiment with the transfected cells performing in red. And as you can see, with this transfection, so that the cells are now back into a position where they can express von Willebrand factor, and with that also generate their own wild Palladi bodies, these cells are back into a situation where they behave like the white bars, which is the controls. They can reseal their membrane, and the calcium release stops, as opposed to the black non-rescued type 3 von Willebrand disease BOECs. And with that, we not only demonstrated the injury to the endothelial cells by complement activation, as illustrated by the calcium influx, but also showed the membrane resealing potential of the endothelial cells. And we think, we're not claiming, we are not naive to claim that this might be the only repair mechanism, but it seems to be a significant repair mechanism via von Willebrand factor release and viable Palladi body mobilization to repair the cells and allow the cells to overcome such an attack and survive. Another little moment of peace before we go into the third and last chapter. So the question now was, while this is a relatively short-term observation, I showed you data basically covering a time window of 1 hour. The question is, what would happen to these cells being exposed to a complement attack over a longer period of time? And the starting observation here was that we needed a readout, and we've thought of motility of the endothelial cells as the starting, as a kind of a meaningful readout, since endothelial cells, being healthy and basically not under stress conditions, are able to close a wound gap that is inflicted. And that's what you can see here on the right bottom. In situations where you have no further insult over time, and this is a 6-hour time gap, the wound that you see in the top at baseline is closing. And if you waited for another couple of hours, then the wound would be gone, and the one layer would be intact again. However, when those cells are stressed, and in our case with complement stressed, then those cells cannot close this wound. So what that tells us is that while the cells seem to be able to repair and to go into some sort of a survival mode, they pay a price, which is is functionality. And one of the functional readouts that is relevant, as I said, is their motility. So I already jumped on motility. You could have asked, could it not be that those cells are just dying, or they're not replicating anymore? We looked into all of these aspects, and we didn't really find a significant difference between the complement stressed versus not complement stressed cells. So we were really left with it must be motility. And there's, of course, a whole literature on how endothelial cells reorganize to become motile, which is fascinating. This cartoon just to kind of hint that this is basically the biology behind. What we thought we should do is we wanted to look at these cells. So this is now live cell imaging of endothelial cells, as you can see, being challenged with an inflicted wound. To the left, the dashed line identifies where the wound border is. And if you, under normal conditions, like non-complement stress conditions, let those cells do what they want to do in those situations, then what you see is what happens. They move towards this wound gap. Over time, they would close the gap. And you can illustrate this even in more detail by basically looking at the cell border. This is live actin and a RAC1 stain, which is involved in cytoskeleton rearrangement. And you see how actively a cell close to the wound border is basically engaging to become motile, reorganize itself, its cytoskeleton, and crawl. If you repeat now the same experiment with complement stress, you see, and this is basically the moment of complement stress is, it starts under normal conditions, the cells move, and then the moment we want to activate complement, we add serum, and it's basically identified by the fact that you see a few red blood cells that are mixed into the serum. So that moment you have complement activated, and almost instantaneously the cells stop moving. And that is very much in keeping with this abrupt and drastic calcium influx that we see with the MAC pore forming. So complement stops, basically makes those cells immotile. And this is the translation into the cell border kind of motility in keeping with this lack of motility. And then the question was, if you let this happen now over a period of time, and this was a 40-minute experiment, is it reversible? So what we did was we basically did an on-off experiment. So let the cells, take off towards a wound. It's again to the left. It's not identified with the dashed line in this case. And then add complement, which is the moment now when you see these flaps and the cells are basically standing still and just standing in one spot. But then take the complement off again, wash the serum off, and replace it with serum-free media when the cells with a little bit of transition period become motile again. So it's an on-off effect over this at least 1 hour or better part of a 1 hour experiment. So, so we were interested in how is this possible? What is happening with the cell to lead to that result? And one of the questions was, what happens? We thought that the answer must be in the calcium, since we felt that this massive calcium influx, you have seen already the left part of this cartoon, must be a major challenge to these cells. So one experiment that we then continued with was to look into the calcium content in these cells over a longer period of time. And on the right side now you see a 4-hour experiment, control at the bottom in black, complement active at the top in red, and then a experiment where we have complement active for 2 hours and then switch to media for another 2 hours in blue. And you see that different from the red line that stays elevated over the entire period of the experiment, when we stop complement activation, in blue, the complement content of those cells normalizes. So that was further support to our concept of its calcium is the driver. And what we thought then was, with calcium being at such elevated levels, we most likely have a negative impact on the mitochondria, and with that, and most likely a problem with the energy household of these cells. And so what we did was we looked at the ATP levels of the cells and found, as expected, over time, this is again a 6-hour experiment in black, a decline in ATP content under complement attack being continuously on. And if we allow it after 4 hours, those cells to recover with serum-free media, so complement was off for the last 2 hours, they started to regenerate and recover some ATP. And we basically measured what you have seen on the left side by measuring the transmembrane potential of the mitochondria. I'll just keep this short now. Could repeat both the stress response where the transmembrane potential breaks down with the lower bars represented by the lower bars, the ones in open bars striped and horizontally striped. The lower bars are the ones where complement is on for various time points. And that reaches the level of the positive controls. And then the far right set of 3 bars is the recovery when you take complement off. And then the question was, how can cells survive this stress period when they have limited or with time exhausted resource of energy and no chance to engage in the mitochondria, regenerate their energy pool. And the hypothesis was then there must be alternative sources for energy. So we thought those cells most likely recruit autophagy to allow for an alternative energy source. Won't be able to go into much of the detail here, but just to orientate you, there is in the end of the or in the center of the autophagy is the autolysosome that allows for the degradation of organelles, proteins, what have you, to basically generate, have different access to generate energy. And what's important for experimental monitoring of this process, there's proteins that are consumed or incorporated and basically consumed on this along this process, including LC3 and P62. And there's chemicals that allow to induce or to block autophagy, rapamycin being an inducer and bufalomycin being a blocker of autophagy. So a little bit of a busy slide, but you hopefully can follow me and see the concept in a moment. So we looked at these proteins, LC3 and P62, expecting that in control conditions, they would be visible. So they are visible. In a situation where we block autophagy, they would not be consumed, they would be visible. And in a positive control situation where we induce autophagy, they would be consumed, as you can see here in the third bar. This is another control where we use both blocker and inducer, and still the proteins are visible since the process is blocked. And now, and this is two of our serum situations that alone don't really have a major effect. Now we use complement activation using our protocol, which is this next situation, anti-CD59 and serum. And you see that, like rapamycin, LC3B and P62 disappear, which identifies that autophagy is recruited. And if you block autophagy, you basically recover both proteins. And the same thing with maybe even a stronger signal appears when you activate complement for not only 1 hour, but for 6 hours. I will skip this here since the, no, I show it to you. And then the question was, this is basically now the induction of autophagy. And the question was then, can we also repeat the finding of, or illustrate the finding of recovery of the cells by basically disappearance of autophagy when the cells recover. So again, controls, media control, rapamycin, and as the inducer of autophagy. And then we apply our protocol, CD59 and serum. The proteins LC3B and P62 disappear. And then we allow those cells to recover for 2 hours and the proteins recover. And if you allow them to recover after 6 hours, you see even a stronger recovery effect. So we can not only show that complement injury or induction leads to this energy deficit and recruits autophagy, but also it turns autophagy is turned off when you allow the cells to recover and allow the mitochondria to engage again to generate energy. The normal way if you want the oxidative way. So complement causes long-term increase in intracellular calcium, which results in an energy deficit of the endothelial cells and a loss of functional aspects of the cells, including motility. However, those cells, the survival of these endothelial cells is enabled, and autophagy is one of the components that allows for that, which is a reversible effect. And that is the end of my story from the lab. And I just end with a couple comments in terms of perspectives. Where is this going towards? I mean, these were hopefully for you in a meaningful way connected chapters of this interest in the consequences of complement activation of endothelial cells, which is a relevant key phenomenon in the thrombotic microangiopathy disease spectrum. What we think we have generated is data that not only contribute to a better understanding of that process, but also could be phenomena that are transferable to other tissue and play a role in tissue homeostasis, in the cellular homeostasis and tissue homeostasis in a more generic, in general way. I think we have identified that the TMA presentation includes not only the endothelial cells, but an intimate interaction with platelets and neutrophils as well. And obviously, I jump to the last point. These insights into a central role and maybe generalizable, transferable role of complement in cell and tissue homeostasis really point towards a future potential for complement blockers or modulators for, of course, for specific diseases, but also maybe in a more generic and a more general way for restoration of disturbed cell and tissue homeostasis. And that's now really it. Thank you so much.